Dear viewers, Assalamu alaikum. In this video, we will study total protein contents measurement in our food sample. The method we use will be Krishna et al., which is published in 2014. The aim of the study is to estimate the total amount of protein by Lowry's method. The principle of this method is when blue color is developed by the reduction of phosphomolybdic and phosphotungstic components present in Folin-Ciocalteu reagent by aromatic amino acids of protein, the blue color of copper complex with peptide bonds of the protein by the Biuret reagent will also be observed at 660 nanometer. The apparatus used for this experiment is test tubes, pipettes, mortar and pestle, vortex mixer, which is called vortex flux apparatus, which is shown on right side of our presentation, and the spectrophotometer. First, we will study how we can prepare phosphate buffer. So if we have to make 0.2 molar phosphate buffer solution of pH 7.2, which is important and mostly used for storage and making protein solution, in which the solution A contains basic sodium phosphate, which will act as weak acid, while solution B, which is 0.2 molar dibasic sodium phosphate, which will act as conjugate base or salt of this weak acid. By using henderson-hasselbalch equation, which is pH is equal to pKa plus log of salt by acid, and the pKa value we used in this sodium phosphate buffer is 6.8, and we have to choose pH of 7.8. By putting these values, we will take antilog and we will get a ratio of salt by acid. So by calculating, we need 28 ml of solution A and 72 ml of solution B, and to make 100 ml of buffer with pH of 7.2. And if we need to make 1 liter of the buffer solution, we will take the same ratio of solution A and solution B. Preparation of Biuret reagent, which is also important in this reaction. Reagent A, we will take 2% sodium carbonate in 0.1 normal sodium hydroxide, and in reagent B, we will take 0.5% copper sulfate and 1% potassium sodium tartrate. We will mix 50 ml of the reagent A and 1 ml of reagent B to make a Biuret reagent. Preparation of Folin-Ciocalteu reagent. Dissolve 10 gram of sodium tungstate and 2.5 gram of sodium molybdate in 70 ml of water. Add 5 ml of 85% phosphoric acid and 10 ml of concentrated hydrochloric acid. Reflux for 10 hours. The instrument for reflux heating is shown in our previous slides. Add 15 gram of lithium sulfate, 5 ml of water and 1 drop of bromine. Then reflux again for 15 minutes. Cool down the solution at room temperature and make up the volume up to 100 ml with distilled water. Preparation of standard solutions. Make stock standard solution of protein by adding 50 mg of bovine serum albumin in 50 ml of standard flask by adding distilled water. So it will make a concentration of 1 mg per ml. For working standard solution, we will dilute 10 ml of the stock solution to 50 ml by adding distilled water, and the concentration will become 200 microgram per ml. Extraction of protein from sample. Weigh 100 milligram of powder sample and grind with 10 ml of the phosphate buffer using mortar and pestle. Transfer sample to centrifuge tube and centrifuge it at 3000 times G for 10 minutes at room temperature. Collect the supernatant and make up to the volume up to 100 ml with distilled water. Estimation of total protein in our sample. Pipette out 0.2, 0.4, 0.6, 0.8 and 1 ml of the working standard solution. Take 0.5 ml of the sample into other test tube and mark as E. Make up the volume of all the test tubes to 1 ml with distilled water. Take 1 ml of distilled water in another test tube and mark as blank. So it will show that it has 0 concentration of the protein. Add 5 ml of the Biuret reagent in all the test tubes, including blank. Vortex the test tubes and allow to stand for 10 minutes at room temperature. Add 0.5 ml of Folin-Ciocalteu reagent to all the test tubes. Vortex the test tube and incubate in dark at room temperature for 30 minutes. Measure the absorbance of blue color at 660 nanometer by using spectrophotometer. Draw a standard curve by plotting the concentration of bovine serum albumin against absorbance. How we can make a standard curve? We have shown in our previous video of how we can make standard curve by using the concentrations of carbohydrates against absorbance. Similarly, here we will use the concentration of standard proteins against the absorbance on y-axis. Calculate the amount of total protein as gram of the total protein per 100 gram of the sample. Thanks for watching. For more videos, subscribe my channel and share my videos. Thanks.